Canal system in phylum Porifera. All the cavities of the body traversed by the currents of water, which nourishes the sponge from the time it enters by the pores until it passes out by the osculum, are collectively termed as canal system. In the Olympus, canal system is seen in its simplest type. In other forms, it may be attain a high degree of complexity, but its general evolution can nevertheless be reduced. But its general evolution can nevertheless be reduced to simple process of growth on the part of primitive olinthos, resulting in folding of the walls and accompanied by a restriction of the colored cells to certain regions. In the gradual and continuous process of differentiation, three distinct types of organization can be distinguished. There are usually three types of canal system meet within the sponges: asconoid type, syconoid type, leuconoid type. Asconoid type. Asconoid type of canal system is the simplest of all the types. In this, there is a radially symmetrical vase-like body consisting of a thin wall enclosing a large central cavity, the spongocoel, opening at the summit by the narrowed osculum. The wall is composed of an outer and inner epithelium with a mesenchyme between. The outer or dermal epithelium, here termed epidermis, consists of a single layer of flat cells. The inner epithelium lining the spongocoel is composed of choanocytes. The mesenchyme contains skeletal spicules and several types of amoebocytes, all embedded in a gelatinous matrix. The wall of the asconoid sponge is perforated by numerous microscopic apertures termed incurrent pores or ostia, which extend from the external surface to the spongocoel. Each pore is intracellular, that is, it is a canal through a tubular cell called a porocyte. The water current impelled by the flagella of the choanocytes passes through the incurrent pores into the spongocoel and through the osculum, furnishing in its passage food and oxygen and carrying away metabolic wastages. The water current in the asconoid sponges follow the following route, that is, water from exterior, then incurrent pores, then to the spongocoel, then to the osculum, and last one to the water out. And the last one is the water out. Asconoid type of canal system is found only in few sponges: Olinthos, Leucosolenia. Here is a diagram of asconoid type of canal system. Syconoid type. Syconoid type of canal system is the first stage above the asconoid type. It is formed by the outpushing of the wall of the asconoid sponge at regular intervals into finger-like projection called radial canals. At first, these radial canals are free projections and the outside water surrounds their whole length, for there are no definite incurrent channels. But in most syconoid sponges, the walls of the radial canals fuse in such a manner as to leave between them tubular spaces. The incurrent canals, which open to the exterior between the blind outer ends of the radial canals by the apertures termed dermal ostia or dermal pores. Since these incurrent canals represent the original outer surface of the asconoid sponge, they are necessarily lined by epidermis. Radial canals, being the outpushing of the original spongeous cell, are necessarily lined by choanocytes and are therefore better called flagellated canals. The interior of the ciconoid sponge is hollow and forms a large spongeous cell, so which is lined by the flat epithelium derived from epidermis. The openings of the radial canals into the spongeous cells are termed internal ostia. The spongeous cells open to the exterior by the single terminal osculum. The wall between the incurrent and the radial canals is pierced by numerous minute pores called prosopiles. The water current in ciconoid sponges take the following route: dormal pores, incurrent canals, prosopiles, radial canals, internal ostia, spongeous cell, osculum, then out. The ciconoid structures occurs in two main stages. The first type illustrated in a few of the calcareous sponges, especially the members of the genus Sycon. In the second stage, the epidermis and mesenchyme spread over the outer surface, forming a thin or thick cortex, often containing special cortical spicules. So these are the diagrams of ciconoid type of canal system. Early stage without cortex and the final stage with cortex. Leuconoid type. As a result of further process of outfolding of the choanocyte layer and thickening of body wall, the leuconoid type of canal system develops. The choanocyte layer of radial canal of the ciconoid stage evaginates into many small chambers, and these may repeat the process, so that clusters of small rounded or oval flagellate chambers replace the elongated chambers of the ciconoid stage. The choanocytes are limited to these chambers. Mesenchymes fills in the space around the flagellated chambers. The spongocell usually obliterated and the whole sponge becomes irregular in structures and indefinite in form. The interior of the sponge becomes permeated by many incurrent and excurrent canals joined to form larger excurrent canals and spaces which lead to the oscula. The surface is covered with epidermal epithelium and pierced by many dermal pores, ostia and oscula. The dermal pores lead into incurrent canals that branch irregularly through the mesenchyme. The incurrent canals lead into the small rounded flagellated chambers by openings still termed prosopiles. The flagellated chambers open by apertures called apophyles into excurrent channels, and these unite to form larger and larger tubes, of which the largest lead to the oscula. The course of water current is dermal ostia, incurrent canals, prosodos if present, prosopiles, flagellated chambers, apophyles, ophodos if present, excurrent canals, larger channels, oscula and out. The leuconoid type of canal system exhibits numerous variations but presents three stages of evolution: Eurypylous, aphodal, diplodal. Eurypylous type. In the eurypylous leuconoid type of canal system, the flagellated chambers are wide and thimble shaped, each opening directly into the excurrent canal by a wide aperture called apophyle, and receive the water supply direct from incurrent canal through the prosopyle. The current of water takes the following route: dermal pores or ostia, subdermal spaces, incurrent canals, prosopiles, flagellated chambers, apophyles, excurrent canals, spongiocele, oscula, then out. This type of canal system is found in Leucilla. This is the diagram of leuconoid type of canal system with uripylous chambers. Aphodal type. In the aphodal leuconoid type of canal system, the flagellated chambers are small and rounded. The opening of each flagellated chambers into the excurrent canal is drawn out into a narrow tube, usually not of great length, termed aphodos. The relation of the flagellated chambers to the incurrent canals remain as before. The route of water current is as follows: dermal pores or ostia, subdermal spaces, incurrent canals, prosophiles, flagellated chambers, aphodos, excurrent canals, spongiocele, oscula, then out. This type of canal system is found in Zoodia and Stellata. And this is the diagram of leuconoid type of canal system with aphodal chambers. Diplodal. In some cases, there is also a narrow current tube, the prosodus, between the incurrent canal and the flagellated chambers. Such a condition is called diplodal. This type of canal system is found in Oscarella, Spongilla, etc. The current of water takes the following route: dermal pores or ostia, subdermal spaces, incurrent canals, prosodus, flagellated chambers, aphodos, excurrent canals, spongiocele, oscula, then out. This diagram shows leuconoid type canal system with diplodal chambers. Ragone type. This type of canal system is found in Demospongi, which in turn arises by direct rearrangement of the inner cell mass. The ragone type of sponge has a broad base and it is conical in shape with a single osculum at the summit. The basal wall is termed the hypophyre, which is developed by flagellated chambers. The upper wall bearing a row of small over-flagellated chambers called spongiophyre. Spongiocele is bordered by oval flagellated chambers opening into it by wide apophyles. Dermal pores or ostia open into subdermal spaces which extend below the entire surface of the body. Branching incurrent canals lead from the subdermal spaces into small flagellated chambers which have been formed by breaking down of radial canal. The flagellated chambers are lined by choanocytes lead into spongiocele. The spongiocele opens by a single osculum. The course of water current of water is ostia, subdermal space, incurrent canals, prosophile, flagellated chambers, apophyles, excurrent canals, spongiocele, osculum, then out. This is the diagram of Ragone type of canal system. Functions of canal system. The canal system helps the sponges in nutrition, respiration, excretion and reproduction. The current of water which flows through the canal system brings the food and oxygen and takes away the carbon dioxide, nitrogenous wastage and feces. It carries the sperms from one sponge to another for fertilization of the ova. Canal system increases the surface area of sponges and increase the volume. That is why the ratio of surface area and volume remain fixed. So that's all about our canal system in phylum Porifera in sponges. Thank you.